This section of notes is on DNA's job of protein synthesis, which has 3 stages: transcription, RNA processing, and translation. So some essential vocabulary that we'll need to know for protein synthesis: gene, which is a part of DNA that controls a trait by controlling the production of a protein. Polypeptides is another term for proteins. Amino acids are the subunits or the pieces that build proteins. And there are 20 different amino acids that are required by living things. Peptide bonds are the type of bonds that hold the amino acids of a protein together. Ribonucleic acid, or RNA, is DNA's helper in protein synthesis. Since DNA is inside the nucleus of the cell, but proteins are made at the ribosomes, which is in the cytoplasm, DNA needs a helper that can take its message from the nucleus to the ribosome so the protein can be made. RNA has some similarities to DNA, but also some differences. So in terms of the sugar, we know that DNA has deoxyribose. RNA also has a sugar, but it's called ribose. So just like DNA gets its name from the deoxyribose sugar, RNA also gets its name from the sugar ribose. The shape of DNA is a double helix, 2 strands that wrap around each other. RNA is single-stranded. The bases in DNA, G and C, are complementary to each other. A and T are complementary to each other. The bases in RNA are very similar. However, there is no thymine in RNA. Instead, there is a substitution of the base called uracil. Now, the bases pair pretty much the same way. G pairs with C, and A pairs with U, as it would normally with T. DNA is extremely long, while RNA can be short. DNA has to stay within the nucleus. RNA has the ability to leave the nucleus through nuclear pores. And then you can see the comparison of the diagrams between the two. When it comes to RNA, there are 3 different types of RNA, and they all have a very specific job in protein synthesis. First, we have mRNA, where the M stands for messenger, and that's exactly what mRNA is. It's a messenger that's going to take DNA's information from the nucleus and bring it to the ribosome so the correct protein can be made. tRNA, the T stands for transfer. tRNA is going to bring the amino acid to the ribosome to assemble proteins. So think of T for transfer because it's going to transfer the amino acids from the cytoplasm to the ribosome. And then finally, rRNA. R stands for ribosomal RNA. And rRNA is a building material. rRNA makes up ribosomes. And this is pretty much the only time that we're going to talk about rRNA. So you need to remember that it's a building material to build a ribosome. We're going to talk a lot more about mRNA and tRNA as they have some specific jobs throughout protein synthesis. So this is just comparing the 3 types of RNA: messenger, ribosomal, and transfer. So the overall process of protein synthesis is producing proteins at the ribosomes using DNA's instructions. The 3 stages are transcription, processing or editing, and translation. So starting with transcription, DNA's message for one gene is copied into RNA, and this occurs in the nucleus. Transcription means to copy, and so that's what happens in the process of transcription. We're copying DNA's information into the molecule of RNA. So the first step of transcription uses the enzyme RNA polymerase. RNA polymerase will bind to a promoter region on DNA. The promoter region signals to RNA polymerase, this is where the gene starts, this is where you need to start copying. Second step, the enzyme DNA helicase causes DNA to untwist and unzip. Remember, DNA has the hydrogen bonds connecting the complementary bases, so it's very easy for DNA helicase to break those hydrogen bonds. In step 3, RNA polymerase begins to add complementary bases to one template strand. So even though we have the 2 strands of DNA open, only one of those strands is actually going to be used to match complementary RNA bases. That's the template strand. By matching up with the template strand, what's actually happening is that a copy of DNA's unused strand is being made. And you can see that in the diagram below. The upper strand that starts with TAC, that is our template strand of DNA, and we're matching complementary bases to it. T in DNA matches with A in RNA. A in DNA matches with U in RNA, because remember, there's no T's, and so on. And if we look at what we end up with mRNA, AUG, CGA, and so on, we're copying the exact message of that unused strand that's on the bottom, substituting in the use for the T's. In step 4, RNA polymerase continues to add complementary RNA bases until it reaches something called the termination sequence. The termination sequence tells our RNA polymerase this is the end of the gene. So then RNA sequence detaches from DNA. It's now called pre-mRNA or primary RNA transcript, and the DNA closes. So this entire process happens within the nucleus. After transcription, after we have the pre-mRNA or the primary RNA transcript, the next step is the editing step, which is also called RNA processing. This also occurs in the nucleus, and it's what has to happen to the pre-mRNA before it's able to leave. So we first have these introns. The introns in the pre-mRNA sequence are copied from non-coding DNA. Think of these as interfering or interrupting DNA's coded message. And because they're getting in the way, they have to be removed or cut out. Exons are the expressed part of DNA's coded message. So this is the information we want to keep, and these are going to be spliced or glued together. So you might think, why do we even have these introns if we're just going to cut them out and remove them anyway? And scientists think that by having the introns, it could be that we have possible alternate splicing methods where we're able to produce different proteins from one single gene. So here we're seeing a sequence of DNA. with some exons and some introns. In our pre-mRNA, we're going to have to cut out those introns and then splice the exons back together. Here's another way of thinking about it, where we've got our exons and our introns. We cut out the interfering introns, and we keep the exceptional exons together. And here's what we're talking about in terms of that alternative splicing. If this is our DNA that we're starting with that has lots of different pieces of information, maybe our mRNA is going to splice this way, which would make one particular protein. Or maybe it's going to splice the next way, which would give us a different protein. Or maybe it'll splice the third way, which would still give us another protein. So we can get 3 different proteins from this single DNA sequence, depending on how we cut introns and splice together exons. So once we have spliced our mRNA, it's no longer the pre-mRNA or the primary RNA transcript. Once we have done that editing process, this is full mature mRNA. And that's ready to leave the nucleus and go on to the next step, which is translation. So in translation, this is when we're going to make proteins at the ribosome. It's called translation because we're translating DNA's message. We're translating the genetic code, or we can think of it as we're taking the mRNA language and we're translating it into amino acid language. In terms of translation, we also have some essential vocabulary. Codons are code words. A codon is a sequence of 3 bases, and it codes for one specific amino acid. So one codon, one code word, is the code for one amino acid. There's a total of 64 different combinations of bases to give us 64 different codons. And so you can see a couple of examples of codons that we have on this slide. tRNA is another important vocab term. Remember, tRNA stands for transfer RNA. There are 64 different tRNAs. Their job is to bring appropriate amino acids to the ribosome to build the protein. tRNA has a double-sided structure. One side has an anticodon. The anticodon is a sequence of 3 bases that are complementary to mRNA codons. The other side of the tRNA is the amino acid side. And so this is the side that's carrying an amino acid, and each tRNA can only carry one specific amino acid. So here is an example of what tRNA's molecular structure actually looks like. And then we can just think of it more in this simplified structure. So now the steps of translation. So the edited mRNA is going to leave the nucleus through a nuclear pore. And then the mRNA is going to attach to a ribosome that's either just free floating in the cytoplasm, or it could even be a ribosome that's attached to the endoplasmic reticulum. The first mRNA codon is always AUG. AUG is considered the start codon because every strand of mRNA that's made is going to start with AUG. So a molecule of tRNA with the complementary codon UAC matches up with the mRNA. And this tRNA will be carrying the amino acid methionine on the other side. So the anticodon is UAC. The amino acid it's carrying is methionine. The The mRNA continues to be read in these 3-lettered codons, and tRNAs continue to bind their anticodon to the mRNA's codon to bring the correct amino acids. This process continues until a stop codon is reached. So there's 3 specific codons that are stop codons, and these signal we're done making the protein. So once we reach a stop codon, the protein detaches from the mRNA. The mRNA then detaches from the ribosome, and the mRNA then is broken down. Since the mRNA is broken down, if the body ever needs this specific protein made again, it must start the process all the way back at transcription in the nucleus, and we would need to make a brand new mRNA. So translation, we're going to translate mRNA into amino acids, thus building a protein. So this is showing tRNA attaching onto an mRNA strand and building a protein. You can see that in red. So in summary, the process of protein synthesis has the 3 stages: transcription, editing or processing, and translation. Translation or transcription occurs in the nucleus. It copies a gene, which is DNA's code for a protein, and it makes the primary RNA transcript, also called pre-mRNA. Then editing also takes place in the nucleus, and that's when introns are removed and exons are spliced together. And then finally, translation takes place in the cytoplasm at a ribosome. The mRNA codons are translated by tRNA anticodons, and amino acids are assembled into proteins. So this is just showing the stages where we have DNA being transcribed into mRNA, mRNA translated into amino acids, where each codon specifies one amino acid. Here again, same thing, just in another sort of a diagram. So now that we understand how this process occurs, we need to be able to translate the mRNA codons into amino acids. Whenever we're using one of these charts, we will always use the mRNA to translate, just like in our bodies, the mRNA is actually what is translated to determine what the amino acids are. So the square chart is one option that you have. To use the square chart, you'll find the first letter of the codon in the far left column. You'll then find the second letter of the codon on the top row. You'll find the box where those 2 letters intersect. Then you'll find the third letter of the codon in the far right column. It must intersect with the box you identified in step 3, and that should give you the name of the amino acid. The other option for mRNA translation is using the circle chart. For this one, we find the first letter of the codon in the center of the circle. Working off the quadrant identified by that first letter, we'll then find the second letter in the next outer ring. And then working off the small slice that was identified by the second letter, we'll find the third letter in the outer ring. And then that will give us the name of the amino acid. It shouldn't matter which chart you use. whether you use the square chart or the circle chart, you should get the same answer either way. Now, the reason that I want you to be familiar with the use of both charts is because when you have standardized testing coming up, when you have our MCA science test in the spring, or when you take the ACT or the SAT in terms of college applications and college admissions tests, there'll probably be DNA questions on those tests, and you're not sure which chart that they might have available for you to use. And so this way, you're at least familiar with both charts. For us, to me, it doesn't matter which chart you use. When I expect you to have to do this, both charts will be available to you. If you haven't watched these 2 short videos on how to use the charts, I would recommend going back into the slideshow and watching those and seeing examples of how they're actually used.